Phew, there we go. I thought for a moment there we were all meeting in the Canadian Rockies. And, well, to be quite honest, I didn't really want to come back to the classroom. But here we are, and it's Chapter 9, Cellular Respiration. So if you'll open your books, we're on page 164. Now, in this section, we're going to give a brief overview of cellular respiration, starting with energy flowing into our ecosystem. As you know, all energy that flows into our ecosystem comes in as sunlight. And without sunlight every day, we would actually run out of energy because energy is not recyclable as matter is. Photosynthesis is going to generate not only oxygen, but it's also going to generate organic molecules to feed consumers. So here we have a picture from your book. We notice light energy coming in. It hits the chlorophyll in the chloroplasts of autotrophs. Those autotrophs are going to take in, look at the 9 o'clock position, they're going to take in carbon dioxide and water, and literally the energy from the sun is going to take apart the carbon dioxide molecules. It's going to take apart the water molecules and literally put them in a different configuration, and so that in the end we end up with organic molecules and oxygen. Now, when we talk about organic molecules, we're talking mostly about carbohydrates, lipids, proteins, and nucleic acids. But during this chapter, we will mostly refer to glucose as our product, and we will follow glucose through cellular respiration. Now, the organic molecules and oxygen will go to the mitochondria, and even before it gets to the mitochondria, aerobic cellular respiration starts with glycolysis. Once glycolysis is finished, then the process goes inside the mitochondria where the rest is finished. Now, from the organic molecules, we are going to reap ATP. Glucose is much too big energy source for the cell. It would be like a bonfire to people who were trying to light a candle. So we've got to break the glucose molecule down into itsy bitsy tiny little units we call ATP. ATP is the energy for the cell. ATP comes out, and of course waste products come out as carbon dioxide and water, and thus the cycle begins again. Okay, we remember that cellular respiration is a catabolic pathway. We are going to be clipping down organic molecules such as glucose, and catabolic pathways are generally exergonic. We remember that exergonic gives off energy. This is going to give off energy as ATP. Now, we have 3 types of processes which we could use. During this chapter, we are going to refer mostly to aerobic cellular respiration, which requires oxygen. But there's also fermentation and anaerobic respiration. Now, in honors biology, we lumped fermentation and anaerobic respiration together into one entity. Actually, they're not the same. Fermentation is a very small partial degradation of your organic molecules, such as glucose, and without the use of oxygen. It only gives you 2 ATPs. Anaerobic respiration, however, goes through glycolysis. It'll go into the mitochondria, carry on citric acid cycle and the electron transport system. But instead of having oxygen as the final electron receptor, it will have other things such as sulfates and nitrates. We will be interested in this chapter mostly in aerobic cellular respiration, which uses oxygen to yield ATP. The equation to cellular respiration is seen below. We've got glucose, C6H12O6, plus our 6 molecules of oxygen. And we will break most of that down within the mitochondria, although glycolysis takes place in the cytosol. Coming out of that reaction, we'll have our six carbon dioxides, six waters. Both of those are waste gas. What we're really interested in is the ATP. For one molecule of glucose, we get about 36 to 38 ATPs. And of course, heat comes out. Now, as we know, heat is waste, and that waste will dissipate into the atmosphere, and we cannot reutilize it or we can't make use of it. That's part of the inefficiency of the process. Okay, if you'll remember back to chemistry last year, you surely studied redox reactions, oxidation and reduction. Oxidation and reduction simply refer to the transfer of electrons during chemical reactions. Now, throughout cellular respiration, you have constant redox reactions. And I might also remind you, wherever you have oxidation, you're also going to have reduction. So what do these terms mean? We remember that when we oxidize a molecule, we have taken away electrons. So it's referring to glucose in cellular respiration. We're going to oxidize glucose by taking electrons away. And when those electrons are taken away, those electrons are going to have to go someplace. Well, the molecule where they adhere or bond to will be reduced. Now you think, if you're gaining electrons, why do we call that reduced? Well, think about it. A molecule that gains electrons will be reduced in its electrical charge. So we are going to reduce a special electron carrier called NADH, NAD+. We are going to reduce it with electrons, and the reduced form will be NADH, and we'll see that in a moment. So the organic molecule is always oxidized, and we will be reducing generally electron carriers. Okay, we have NaCl here. Notice that when sodium comes together, if you'll remember, it has one electron in its valence shell. Chlorine has 7. Sodium would love to get rid of its electron. Chlorine would love to take it on. And so sodium will be oxidized. Chlorine will be reduced. Now, your book shows us this hokey little equation. I actually think it's a decent equation because the X and Y represents any molecule you might have, whether it's an organic compound, inorganic, whether it's glucose or lipids or whatever. Xe- is going to be oxidized. It will lose its electron with a negative charge. Y takes it on, so Y is reduced. Notice that the product of Y on the right side of the equation is now negative charge. So as we go through aerobic cellular respiration, you will need to keep track of what's being oxidized and what is being reduced. I have noticed that on AP tests, they often zero in on small facts like that. Okay, we've already talked about that. On the left here, we see methane. This picture is in your book, figure 9.3. Notice that methane is a hydrocarbon. Remember that carbon and hydrogen bond they have like electronegativity, so they're going to have a nonpolar charge, a nonpolar covalent bonds. Notice that oxygen is highly electronegative, so what is oxygen going to do? Oxygen is going to start stealing hydrogens away from the carbon, and ultimately the methane will become oxidized, the oxygen will become reduced, at least that which is in water will be reduced, and that is what we will see with glucose. Okay, now you will need to know this equation. I'm sure you already know it from honors biology. Oxygen is what's being reduced, and that means it's taking the electrons from hydrogen, and so the end product of cellular respiration is carbon dioxide, our water, and of course our energy. The energy in the form of ATP is what we're after. Now I want to remind ourselves of oxygen's electronegativity. It is one of the most potent oxidizing agents of all molecules. Now think about what I just said. If oxygen will oxidize glucose, oxygen is an oxidizing agent. Glucose will be the reducing agent because it's the agent that will reduce oxygen. So that sounds kind of confusing. If it is confusing to you, read that in your book a couple times. So glucose will be oxidized by oxygen, but oxygen is the reduction agent for oxygen. So let's get this clear one more time. Glucose will be oxidized by oxygen. Oxygen will be reduced. Glucose will be the reducing agent for oxygen, and oxygen will be the oxidizing agent for glucose. So notice that as well. Okay, let's get into the nitty gritty. In cellular respiration, the electrons, like I just mentioned a moment ago, from organic compounds, we're going to zero in on glucose, are usually transferred to a molecule called NAD+. Now, NAD+ is a coenzyme. If you remember coenzymes from honors biology, a coenzyme is generally a vitamin that is used to activate or needed in order for an enzyme to work. So NAD plus is simply a coenzyme. NAD plus stands for nicotinamide adenine dinucleotide, and it's just a coenzyme. But it's also an electron carrier, transporter. It's like an electron taxi. The electrons are going to climb into the taxi of NAD plus, changing them, reducing them to NADH, and NADH will then ferry the electrons to where they need to go. So let's mention this as well. The NADHs, the electron carriers, will pass the electrons to the electron transport chain within oxidative phosphorylation. Now that's a new term we'll talk about another day, but the electron transport chain is within a bigger, broader process called oxidative phosphorylation. Once we get to the electron chain, the first protein in the electron chain is more electronegative than NADH is. It will steal away the electron from NADH, and then, as we'll see another day, each protein within the electron transport chain is more electronegative than the last protein, and thus the protein bebops down the chain where oxygen is waiting. And of course, oxygen, being a potent electronegative oxidizing agent, will grab away the electron. Hydrogen will be waiting there, and thus we will have the waste product of water. Okay, now the question is, why do we need to go through this entire process? Well, look at the panel on the left, and I remind you that as we are reaping energy from glucose, if we reaped it in one big gigantic step, it would be like an explosion of energy. It would be so big to the cell, so hot, so explosive, that it literally would kill the cell. So essentially what we're doing is panel B, cellular respiration. What we're going to do is we're going to reap the energy, the free energy in NADH in tiny little units as we go down the electron transport chain. And each time it bebops down the chain, we will be reaping small amounts of energy in the form of ATP. Okay, the stages of cellular respiration, a preview. If you remember from honors biology, aerobic cellular respiration has 3 stages. We have glycolysis, which breaks down glucose into pyruvate, sometimes called pyruvic acid. It depends on whether it's an ion or not. We'll just call it pyruvate. And glycolysis happens in the cytosol, and it does not require oxygen. Then we have the citric acid cycle, also sometimes called the Krebs cycle. The two pyruvates from glycolysis will move into the mitochondria where the citric acid cycle continues to oxidize glucose, reducing NAD pluses to NADH, where they will go to the final stage, oxidative phosphorylation. Now, oxidative phosphorylation we did not talk about in honors biology, but AP biology just gets a little bigger and a little deeper. Within oxidative phosphorylation, we have two stages. One of those is the electron transport chain, and the other is chemiosmosis or chemiosmosis, depending on who you listen to. Okay, this is an extraordinarily helpful visual in your book. It's visual 9-6 on page 167. I would recommend you be able to see this illustration in your mind's eye for any test or quiz. If you know this, if you can see this in your mind, you can answer many, many questions from just seeing this visual in your head. So let's look at it. On the left hand side, we have glycolysis taking place. Notice it's outside the mitochondria, it's in the cytosol. We're going to break glucose into two. Glucose is a 6-carbon sugar. We're going to break it down into two 3-carbon sugars called pyruvates. We have partially oxidized glucose, reducing NAD pluses into NADHs, and those NADHs will ferry or taxi or transport the electrons to oxidative phosphorylation and find the electron transport chain. Now, glycolysis also reaps 2 ATP. But remember, ATP isn't worth much. We're only going to get 2 of them, and they're like pennies are when we compare a dollar. So ATPs are very little consequence as compared to NADH. Once we're done with glycolysis, we'll enter the mitochondria where we'll start the citric acid cycle. The citric acid cycle continues to oxidize the pyruvates into smaller and smaller waste units called carbon dioxide and water. We will get a couple more ATPs out of them, but again, what's important is we are going to reduce NAD pluses to NADH, and we're also going to find a new electron carrier called FAD, and that will be reduced to FADH2. There it is, sorry. Finally, we'll go to the oxidative phosphorylation where we will find the electron transport chain and chemiosmosis. Now, I'm not even going to go into chemiosmosis today, but notice what we have here at the bottom right. We get 32 to 34 ATPs out of the oxidative phosphorylation stage. What do we get out of citric acid cycle? 2. What do we get out of glycolysis? 2. Altogether, we should reap somewhere between 36 and 38 ATPs. Okay, we will end there. Tomorrow we will pick up sections 9.2 and 9.3 and probably 9.4. 9.2 and 3, I believe, are glycolysis, citric acid cycle, and then we'll go into the oxidative phosphorylation. My recommendation is for you tonight is for you to read the textbook and understand this to the best of your ability. See you tomorrow.